companies like yours are often innovating. They get a molecule, they sell it, it gets rolled up for a tiny fraction of what it's ultimately worth to the large pharmas.
But you work in the field when you kind of like, you go home and you work for something that is for the really societal benefit, right?
And welcome to the Jimini Health Podcast. I'm Mark Jacobstein, the co-founder and president of Jimini Health. In our conversations, we explore the frontiers of AI, mental and behavioral health, and any interesting ancillary topics from precision medicine to genomics. Speaking of ancillary topics, I am very excited today to have Dr. Yaniv Erlich with us. Dr. Yaniv Erlich is the founding CEO of 11 Therapeutics. He's a former CSO of myheritage.com and is a recovering academic, I think in his own words, former professor of CS and computational biology at both Columbia University, and he was a fellow at the Whitehead Institute. Dr. Erlich has more awards than I'm going to list off here, but we're really excited today to have somebody who has worked on so many different interesting fields here to have a conversation with us. So in prepping for the conversation today, one of the things I did was I read some of your papers, and there are very few people, especially as early career as you are, who have worked in so many different fields. So maybe if you don't mind, we'll take a few minutes, walk me through sort of the journey to where you are today with 11 Therapeutics, and also if there's sort of a common thread you would pull through and how you see sort of the journey to get here, sort of what led you to where you are now.
Yeah. So I'm doing the same thing. I'm a guy of information and DNA. These are my two loves, and I just try to marry them in different intersections of my career. I started my career, I did my undergraduate studies in Tel Aviv University at the program of neuroscience. So I actually learned psychology and biology. And I moved to Cold Spring Harbor to do my PhD. And when I moved to Cold Spring Harbor, I thought I'm going to do neuroscience in Cold Spring Harbor. But we had this little thing called next generation sequencing, and that was the advent of it in 2005, 2006. And pretty much everyone that knew how to code was recruited by people in desperate need to actually go and work on these sequencers that used to spit quite a lot of information. And it's funny because we had this guy, he was a visiting student from Spain, and he sequenced his first library with Illumina. And he used to open his sequencing files in Word, Microsoft Word, copy paste the first sequence, go to UCC genome browser to the blot tool, and search in the genome. And then he wrote it. The first one. First, he tried to open the first library, and that time it was 2 million sequences. It was quite a lot back in the day.
Well, and just to contextualize this for people who aren't in the omics world, in 2005, 2006, as the dawn of NGS, the cost of sequencing and the throughput of sequencing were changing so rapidly, it makes Moore's law look anemic. I mean,
exactly,
and it's not the magnitude sort of coming off the cost. And in every year, it's astonishing charts if you want to go look them up. And yeah.
So if you want to think about it, if you take all the gold output of the US, all the mines in the US, the amount of gold that was produced in 1990, right? It cost more than that to sequence 1 million nucleotides in 1990. Now, back to the current day, sequencing 1 million nucleotides cost around maybe 5 grams of aluminium, right? Something like that. So it's a major change of gold, all the production in the US, to something that you wrap your sandwich like 30 years later or so.
Well, the way I think about that, in the same way that, of course, today everyone is interested in AI and ML, in part because that's where the most interesting things that are happening. Circa 2005, 2006, this was the most astonishing sort of revelation of interesting data that would help us unpack the great biological mysteries of the world. I mean, it's such an extraordinary time to be alive, especially if you're interested in data and coding.
And still today, because if you think about it, all the digital world, everything that you train your AI models, right? The digital world is about 10 to the power of 25 bytes or so. And we call it big data, right? It used to be a buzzword like a decade ago, big data. Now, if you think about all the data that is encoded in our biosphere on DNA, you just count DNA molecules and you treat each nucleotide as 2 bits. It's about 10 to the power of 37 bytes. So that's like the 12 orders of magnitude. It's quite, like much more data exists right now on DNA molecules than on any digital media that you can think of.
Yeah, and it underpins how our CNS works, how the immune system works, all the most how we work ultimately. And so in some ways, it's not just that it is an incredibly rich and important and exciting data set. It is essential to sort of understanding ourselves. So this is 2006 or so. So you get into this project.
So I get into this project. The guy tried to open this like Word document file. Took him 10 minutes just to open the Word document, tries to align it by hand, said like, okay, it should be something, let's do something different. And he came to the bioinformaticians, which is apparently me, because I knew how to code before I joined my PhD lab. And then my PhD advisor at that time, Greg Hannon, told me, you should come to my lab. And Greg is like kind of like a mafia person. He sent some people, actually sent Jim Watson, the Nobel laureate, to come to speak with me and tell me that I should join his lab and I should not make any mistake by declining this offer. And so I joined the lab, and it was quite successful.
Offer you cannot refuse.
Yeah, exactly. It was like, he sent some messengers. It was a quite intense pressure. And I joined the lab, and I worked on first how to get better data from these machines using machine learning. I realized that we have so much data in the machines, and I have some reads that I know exactly in the genome where they come from, so I can create basically supervised learning. It's a classification problem reading DNA. You have some analog signals, you try to put them into 4 classes of A, C, G, and T. So it's a classification problem. And instead of just using model without any kind of like notion about how the error looks like in a specific run, I can train the model on the molecules from the same run that come from a genome that I know quite well where they come from. And then I can fine-tune the boundaries for each kind of like cycle to get precise readouts for the nucleotides. So actually I wrote this supervised learning model. We utilized it, was published in Nature Methods, and we were able to go from 36 nucleotides, this was the limit of the sequencer at that time, to 78 nucleotides. And the reason for this weird number, 78, is because we ran out of disk space. I bought the largest disk space that you could find in B&H at that time. We just ran out of disk space. But we were able, and this was basically at that time.
I can sympathize. I grew up coding on a TRS-80, which actually literally had essentially zero memory, 32K. Anyway, so I can sympathize. And maybe just a little bit of context here for any of our listeners who come from, say, a digital world, not a biological world. I remember because I'd spent 20 years building companies on the IT side, and then I moved over into biotech. Was shocked to discover that biology, everything is flimsy. And you called it a classification problem. I was shocked to learn, for example, that the reads that come off an Illumina sequencer are not 100% accurate, right? That to your point, it is a classification problem as to whether or not it is an A, a C, a G, or T. It is not digital. It is not, you don't know for sure. And that the error correction component of and sort of information systems, like cleanup component of building out these systems is incredibly important. And this is obviously work that it sounds like you were doing in the early 2000s, which is really when NGS was coming online.
Yeah. So then after kind of like we addressed this problem, and by the way, it's interesting, the manuscript was cited hundreds of times, but not so many people used it. First, because it took some time to run these models. It was very, you can build a very good machine learning AI model, but if it takes time to actually like use it in real time and you need to call the bases, most of them didn't want to do that. That's the first thing. The second thing, Illumina solved the problem by using better reagents. So you can be very fancy about the decoding, but if you have better encoding, you don't need to be that smart, right? And I think that's a famous quote. I once saw it in some seminar that you only need to be smart when you run out of data. So if you can generate enough data and you don't need to be that smart. So that's kind of like one fragment, one piece of my PhD. The second one was we wanted to sequence tens of thousands of samples on one Illumina run. But there is a problem, right, Mark? Because I can pull all these samples at once, but then I don't know which one is which. So then I can tag each sample with a specific tag, but the tagging itself becomes so cost prohibitive more than the sequencing. So I kind of like developed this technology that I can pull samples into combinatorial pools overlapping pools. So maybe you and I and another person will be in one pool, then you and this other person, but not me, will be in the second pool. And by looking at the pattern of mutations, I can decode it back. And it's basically a matrix inversion problem, but it's a sparse matrix, so you can actually do it, although the matrix is, the system of equation is undetermined.
Loosely sort of a form of multiplexing without having to do fancy chemistry that is expensive and potentially destructive to the DNA itself.
Yes. And to do it like very cheaply, it was also connected to a CS field at that time called compressed sensing that I utilized some of the algorithm. I realized at some point that I'm actually doing exactly what all these mathematicians are studying, just in the context of DNA, which led to very interesting publications and collaboration with mathematicians that I never thought I would collaborate with. And so that was pretty cool. And so this is kind of like how my career started, thinking about like DNA and information and encode information in DNA. So this is kind of like the way that I started my career.
And it seems like if I could pull on that thread, really your whole career has been about DNA and encoded information, although now we've moved into RNA, et cetera, but sort of downstream. Tell me a little bit. I mean, one of the things I do want to get into here is your work at MyHeritage. First of all, it'd be great for you to describe sort of the company and the project, but specifically, although there's an ancestry component that is fascinating and a historical component and pop health historical stuff that's wonderful. I'm most interested sort of in the health and biological implications of building these giant databases of humans and the way that they're interrelated. Yeah. So maybe talk. We're going to jump way ahead here because I know we skipped over your time at Columbia and what have you. Maybe we can circle back to it. But the work that you're doing at MyHeritage and how that bleeds into sort of personalized medicine that's genomic based.
Yeah. So in genomics, we have two modes where we try to predict a disease, right? One is called Mendelian conditions. Mendelian conditions are affected by one gene, and there is a specific mutation in the gene. For instance, cystic fibrosis is a case like that. There is a gene called CFTR. If the gene has some faulty mutation in it, and in both copies, in the case of cystic fibrosis, you basically develop this condition that is associated with this erosion of the lungs over time and some other types of complications in other organs. And that's one type of disease or condition. And these are mainly affected by one gene with a very large magnitude of effect. And these are relatively quite easy nowadays to predict and to discover those. If you give me an individual with a condition, I can go screen their entire genome very rapidly. I will contrast their genome to the genomes of tens of thousands of healthy individuals, try to find differences, and then I will do a few more tricks, and I'll be able to typically to go and hone on the causative mutation.
Find the pathway, because there'll be a lot of single nucleotide variations in any given human that are maybe perhaps pathogenic, but there'll be ones that are suspicious because they're in the right pathway that might cause the problem.
Right pathway, they also, usually it comes in families, so it will co-segregate with the individuals that are affected. You can do a few tricks to narrow down and make sure that you get to the right gene. Yeah. And then you can do the opposite. If you have a person with the mutation, you can, in some cases, predict that this person is more likely to suffer from the disease. For instance, there is the case of Angelina Jolie, that said that she is a carrier for BRCA, I think it was BRCA1 mutation, that increased her likelihood to develop breast cancer by orders of magnitude. We talk about nearly 90% lifetime risk up to age of 70 to develop breast cancer. So this is one class of diseases or mutations, and quite easy to do these predictions. There are modulators. It's always a bit more messy in biology, but relatively simple. The other type, and maybe I can just open parentheses here because it's fascinating. The way that I was introduced to this topic was actually very, very interesting. I met, there is in the ultra-Orthodox community, the Jewish community, they would not go for abortion. It's against the Jewish halakha in certain cases. And it can create a stigma if you have someone in the family with this rare genetic condition. Now, Ashkenazi Jews suffer from a series of genetic diseases due to this kind of like founder event. We have many people started from a very few people. So
the average relationship between any two, we're probably third cousins. Yeah,
exactly. Something like that. So any married, Any marriage between two Ashkenazi Jews is in the order of third cousin marriage, which increases the likelihood of developing some sort of a genetic disorder. There is a guy called Rabbi Eckstein that he lost 4 kids to Tay-Sachs, one after the other, just horrible, in the 80s. And he decided to do something about it. And Tay-Sachs is a horrible disease. The kids, they're born healthy. Everything is fine. After 6 months, 8 months, they start to lose, due to deterioration of neurological circuits, they start to lose skills. Suddenly, they don't sit unassisted anymore. They become floppy, suffer from seizures, go blind, deaf, and horrible death after 4 years. No
neurodegenerative diseases of these kids are just the work. My wife happens to work on lysosomal storage disorders, and they are some of the most terrible diseases.
Terrible disease. So he watched 4 of his kids suffering through that, just horrible. And he started this organization called Doya Sharim, which goes to yeshivas and schools when the kids are about 18 years old. And they offer to do carrier screening. So they will test your DNA. They will not give you the results back, Mark. You cannot get the results. What you get is a 6-digit ID number, and that's it. Now, typically in this community, you go through arranged marriages. You don't go to Tinder. One of the very first steps of the arranged marriage is that the families exchange a 6-digit of the wannabe bride with the wannabe groom. Now you can call to the hotline of Doya Sharim, put your 6-digit, the 6-digit of the bride, and it tells you advisable or inadvisable for this marriage before even you go
so
to
two Tay-Sachs carriers are inadvisable.
Exactly. And for other types of diseases. Now, if you're a carrier for cystic fibrosis and your future wife for Tay-Sachs, it's fine because these are recessive diseases. So they were able to eradicate Tay-Sachs from this community. Nearly no cases at all since they started. And so kind of like you talk about personalized medicine, but this is a way, and these are some of the most conservative people on earth that I met. Right? Super, super ultra orthodox, right? And you don't need fancy technology sometimes. You just need to build the technology, to build a product for the people, for what they need and what the community needs. And they're able to just nail it. And I don't think there is anything in comparison for genetic screening. So that's one component of giving genomic information to people.
Yeah.
The other type of conditions, we started by talking about Mendelian conditions, are what we call complex traits. Complex traits are affected by many genes, where each of which contributes a little bit to the trait. For instance, height. Height is 80% of height variance is explained by genetics, so it's highly heritable trait. And it's not affected by one gene typically, but your height, under normal conditions, not talking about some rare genetic disorders, height is determined by many, many genes. Each one contributes another millimeter, 1 minus millimeter. It will just add up to give you basically the height that you're now. Many diseases are like that. Also, risk for heart attack, BMI, psychiatric disorders. Most of them basically operate this way. Many tiny mutations in the genome, each of which contributes a little bit. Now you can think, okay, that's a fantastic problem for machine learning, right? Because the genome has 3 billion letters. We have millions of variations.
What we need, of course, are the phenotypes. We need at least a reasonable phenotype of the patients, and then enough genotypes to start to pull out the polygenic interactions. Yeah. It's interesting because I've had a lot of conversations over the years with people who sort of wanted to work on this problem. I don't know, you may know Matt Rabinowitz, who is the founder of Natera, for example, started a company called Myome. And I talked to Matt back in the day about how he was running Myome. And the trick has always been you need enough genomes, and either whole exome or whole genome. You need enough phenotypic information, and then you need patient consent, which of course is critical and very important. Although most patients, in my experience, are actually very excited and interested, particularly if they happen to have a chronic or debilitating disease. But even if they don't, generally interested in sort of contributing to science. But that process of securing the consent is also a really, really critical piece. And I'm curious as to how that ties into what you were doing with MyHeritage, because I've always thought of something like MyHeritage or 23andMe as amazing sort of hacks to get at people's genomes and phenotypes in order to unpack some of these biological mysteries. But I don't know if that's sort of how you felt.
So I think the problem, I think, with kind of like direct-to-consumer genomics in general is that first you get the people are too healthy, right? If you have the means and the energy and the time to go to do direct-to-consumer test, you're not suffer from some very extreme condition in most cases.
Right.
Or at least when we sample the population, we get people that are on average more healthy than the average kind of
and probably highly educated, affluent. It skews
affluent, highly educated.
Yeah,
especially those that consent for research. So that's one tragedy of this endeavor. The second one is that we rely on self-reporting information. And maybe another thing is that people that typically come to these companies that are in, let's say, 40s, 50s, many of these diseases manifests at later ages, right? 70 or so. And if the disease was fatal early on, I will not see them. Right. So there is some sensory effect there. And one way that we can overcome it, Joe Piquet, that used to be at Columbia University with myself, we had this manuscript where we show that you can ask people about the health of their parents. And since you share 50% of your genome with your parents, so 50% of the features are correlated, you can still utilize that to discover some things about the parental, kind of like the association between genome and phenotypes based on just asking about the parents. By the way, we found that asking women is better than men because it seems that they have better recollection. You can get better. You see the R squared that we got from the females was just better in the way that we could predict.
Fascinating.
So, yeah.
Is that a Y chromosome problem? Is that women do a better job of actually describing the phenotypes and the histories?
The parents take better. When my mom called me about I suffer from this and that, I was like, okay, whatever. It's like if it was like a daughter, maybe she would pay more attention about that, something like that, right? But also it helps with not just with age-related diseases, but if you think about it for sex-specific conditions, I can ask males about whether their parents or their sisters had breast cancer, and even if I don't have the sister or the mother, you'll be still informative because if, let's say, your mother had breast cancer, 50% of the genome is correlated, and now suddenly I can collect much more data. Right. So that's a trick that we showed can work. The second maybe challenge when you do this type of direct-to-consumer tests is that people on the internet, they tend to, the information is not as accurate as maybe a medical health record. We saw it with height. You ask people about their height, you expect something quite smooth along the range. Let's talk about centimeters, right? Not inches. You expect, people should be like.
I was a high school basketball player. They used to list me at 6 feet. I am not 6 feet tall. We listed everybody an inch or two taller than they actually are to intimidate the opposing team before we would go play.
You always want. So if you look at the last digit of the height, it should be distribute uniformly, right? Like between 0 and 9 should get the same probability. And it's not. 0 and 5 are very prevalent. People like to, and it's round up. Nobody rounds down.
Everything rounded up. Yes, sure.
Yeah. So and sometimes you have that. So it can be more challenging to get that information from people online. One thing that we're able to see is that we at some point we try to see how long it takes people to answer the questionnaire. Just the time. And we found that the time itself can be a phenotype. How fast are you, cognitive ability? And we saw actually a decline with age for that and some association with known Alzheimer markers, although these people don't suffer from Alzheimer. That was another thing. So you can try to sometimes trick it to get information that will be more reliable indirectly than asking the question.
Yeah.
But it can provide you quite a lot of data, this direct-to-consumer, but I have to say there are other fantastic ways. There is the UK Biobank these days that has 500,000 people, deeply phenotypes, well curated, so that's another option for you.
Yeah, Nick Wagley, when he was over at Dana-Farber before he went over to Genentech, had the Metastatic Breast Cancer Alliance project where he recruited metastatic breast cancer patients through sort of social means. It's very interesting because when I look at these sort of larger population health size studies where you're trying to find polygenic interactions and you need sort of good phenotypes, especially if you're talking about people with a particular condition, they're highly, highly motivated populations, right? If you are a woman with metastatic breast cancer and you're hoping for there to be a breakthrough, and all you really need to do is give consent for your EMR information and maybe a few blood draws, it's a pretty small lift given the research upside and whether you're thinking about yourself or for your kids, et cetera. I'm generally bullish on some of these approaches, and at least my personal opinion on this is we need both. There are problems with the 23andMe's, the D2C sort of approaches that you outlined, and I think they're very real. Still, even if they're noisy, they can be so broad that I think they can uncover certain things. And then you have the much, much more specific. I've got a community of people with MPS II, and there's only a few thousand of them in the entire world. Yeah. But we're gonna whole genome sequence every single one of them, or actually even a few dozen of them might be all you need to find very, very interesting, polygenic stuff.
Yeah, and at some point, for certain diseases, the question is if we're going to exhaust the population, basically. Right, we're going to exhaust every possible case on Earth, and still you don't have enough statistical power to detect all these different modulators or interactions that you want to get.
Well, at the risk of jumping ahead to your what you're working on right now in RNA, I mean, one of the things that has struck me, I worked at Guardant Health years ago, where we did circulating tumor DNA sequencing for metastatic cancer patients, and naively, because I wasn't from biotech when I first came into the company, I assumed that we're going to sequence hundreds of thousands of metastatic cancer patients. We're sort of going to unlock all of the mysteries of cancer. And then I realized, of course, that while the DNA is important, it is but one component. Of course, you have your genomics, you have all the other omics, you have the transcriptomics, you have the proteomics, you have the immunomics, and sort of which layer in the biological stack to be looking in order to uncover the mysteries and or to be intervening in order to be developing therapeutics is a really interesting and hard question. And I'm curious, given sort of the history with DNA, I don't know how much you talk about what you're currently working on. Your website is certainly fairly stealthy, but I'm curious if you don't mind talking about sort of that journey to RNA.
Sure, I will, but I think in general, if you want to understand biology, I would argue DNA is the best molecule to understand biology. And the reason is, there are a few reasons. The first one, I can read it with extreme precision compared to anything else, right? I have high throughput sequencing. It's commodity, it's cheap, it's reliable, it's digitized, fantastic. So that's the first reason. Second reason, even more important, it's the error of causality. DNA precedes everything. That's the first thing, when you are conceived, first it's DNA, then everything else. So, and you get your genes, you get them using this Mendelian randomization from your parents. Your parents don't select, I give this gene, not this. They give it random, right? So there is some randomness event here that randomizing over. Not yet, yeah.
Not yet.
That's Gattaca type of approach. So that's another thing, right? It's randomized, so you get that. So that's the second reason. Third one, I can, in many cases, with certain accuracy, I can impute the expression of other omics data from DNA. So if I learn enough, you give me enough kind of DNA plus another data set, I can start look at the DNA and say, okay, let's impute the other data set with certain accuracy. So I'm not saying this is of course, many people probably would hate me after that saying that, but it's
not all the answers, but it's a lot of the answers.
It's a lot of the answers are already there. And then you can always argument it, but that's a very strong starting point. Yeah. And that's the reason why we have human genetics and we don't have, human proteomics as entire field by itself.
The readability and stability piece is incredibly important. I'm reminded of the old joke about the person who's looking for the dollar bill underneath the light, and why are you looking there? It's because where the light is. But just for folks who come again from don't come from sort of the omics world, you made the point earlier. It's much cheaper and easier to do high throughput sequencing of DNA than it is of all the downstream things, RNA, proteins, et cetera. People are working on high throughput RNA and proteomics, et cetera, but it's much harder, much more expensive, much more flumpy and inaccurate.
And there is a very simple reason for that. The simple reason is that DNA can replicate.
Right.
And all these reading methods, if you can replicate, becomes much, much easier. If you cannot replicate, you just destroy by measurement. It's very hard.
Right. And the other component, which, again, I didn't appreciate until I started my time at Guardant, was just how stable DNA is, which of course it is if you think about its role in our historical evolution. Like that's part of why DNA is it's extraordinarily stable. And I know you've done a lot of work on DNA storage, which is incredibly interesting, and hopefully we'll have time to get into. But the story I'll relate is when I was at Garden, we used to talk about, so we would get 2 tubes of blood FedEx to us from somebody, and they weren't stored in any special way. And they literally 2 tubes of blood, which sat on a tarmac in Memphis, Tennessee, which is where FedEx has its headquarters, in the middle of the summer, and arrived in our office the next day, could be sequenced accurately. And the DNA had suffered in no way. Now, that was not true for all the, like, if you were trying to find proteins or RNA or anything else interesting in the blood, but for the DNA, it was in great shape. And that's incredibly, it's a wonderful, fortuitous thing for genetic researchers.
And since you're interested in kind of like machine learning and genetics, we're talking about that. One thing to kind of like keep in mind that genetics and statistics started hand in hand. Ronald Fisher from F-Test, right? He coined the word heritability, and he also invented the word variance. And this was in the same manuscript in 1918. The first time that someone used variance, that someone said, let's take standard deviation and let's square it. So then we get kind of like, and it's weird because the units becomes squared units, right? Of like you measure the average and then squared units of the average. He did that because he wanted to be able to parse variation and to look at the genetic component of variation, which is heritability. So he invented that term in the same manuscript that heritability was invented. And so that's kind of like one, and he was a statistician, but also a geneticist. Pearson, from Pearson correlation, he wanted to do correlation to look at the height of parents versus offspring, and regression to the mean means that the offspring tend to regress to the average of the 2 parents. So many of these things were
with me. I have a very tall father. It didn't work out.
So that's kind of like these fields started together, genetics and statistics, which
That's fascinating. I don't think I knew that.
Yeah.
So maybe just to talk about 11 therapeutics, then you are working on RNA therapeutics, though, and I'm curious about sort of how much you can talk about that and sort of the relationship between that and some of the genetics work that you'd been doing earlier.
So we try to make RNA DNA again, in a way, right? And if you think about it, RNA therapeutics is amazing, right? We saw it with COVID vaccines, but the biggest problem, and it's safe, and you can create beautiful kind of, the COVID was tested in billions of people, the COVID vaccine. RNA is unstable molecule, as you described, right?
Right.
And if you want to treat people with chronic diseases, if you give them RNA molecules, they will maybe create an effect, but it will be very short-lived, this effect. So you want to stabilize the RNA molecules. Now, DNA, which is only one change. There is only one atom change between RNA and DNA. That's the oxygen on a specific location of the sugaring of the.
It's the deoxy.
Exactly. You remove the oxygen, and suddenly it becomes very stable. But there is a problem. If you just remove the oxygen, it will not translate into a protein. The ribosome, the machinery that reads the RNA to translate into protein, it doesn't like to get DNA. So we at 11 Therapeutics, we are interested in creating RNA molecules that, on one hand, have these chemical changes, and I can do more changes, just add or remove an oxygen, I can do many more changes. So the RNA molecule, it will stabilize the RNA molecule, the change will stabilize it, but will still enable the ribosome to read and to produce my protein of interest. So we use chemical modifications to the RNA molecule in order to stabilize it. And that's kind of like what we are working on.
That's interesting. And the goal here is you need the RNA to be stable because in the context of, say, a COVID vaccine, it's okay that the mRNA is short-lived because all you need to do is stimulate an immune response. We need the mRNA to go in, make a make a spike protein that your immune system recognizes as foreign, generates a response, and now your body is sort of primed for when real COVID arrives. And it's okay. In fact, it's probably good that the mRNA is then essentially flushed out and gone very, very quickly. Downstream effects are limited. Very different in a scenario where you want the RNA to stick around and continue to produce a protein an enzyme of some sort that is beneficial for somebody with a disease. Are there particular disease areas you're going after at first? Is this oncology, autoimmune?
It's actually, we are a platform company, so we're not that interested in specific. We have some internal programs right now, but we think that this technology can go to different types of indications in general.
I see. So the goal is to build a generalized platform, understand how to, I'm going to call it up armor these mRNA molecules to sort of perform what you want them to perform, to be as stable as they need to be, yet still able to express. And then you can downstream license this to any biopharma company that's producing whatever it is they're trying, whatever enzyme, whatever protein they want to produce, ultimately via these enzymes can license the platform.
Yes. And what we found is that it's not that simple. There are some molecules that like this modification, some another modification. So now we're actually building this, trying to decode the structure-activity relationship of how to actually produce the molecules. So that's some of the stuff that we're doing.
This sounds like a perfect ML problem.
Exactly, but we need a lot of data, and we're working on trying to develop to get enough data.
This is the conundrum. Well, there's two conundrums of every tech bio that I know. One is, how do I generate in a reasonably inexpensive, high throughput manner the biological data that I need to train my models to do anything useful? And then how do I deal with the fact that in the world of tech bios and biotechs, all of the value accrues to the people who actually make the drugs? And so licensing the platform downstream can work, but is challenging. And you end up with a lot of biobucks deals that take many, many years to sort of to play out. But it's noble work, and it's incredibly interesting and exciting. I love the approach.
It is, but also there is something I think unique in biotech in general compared to tech, right? In tech, first you look at the incumbents in tech. These are not the same incumbents 20 years ago, right? Different companies in general, right? We had like Nokia and the ups and downs, and Microsoft, Intel, IBM, and today totally different companies.
Today it's NVIDIA, yes.
Yeah, NVIDIA, right? The NVIDIA of the world, and Google, Facebook, 20 years ago was like a quite small company, I think, at that time. You look at pharma, right? 20 years ago. These companies, the top 10, still the top 10, almost still the top 10. And, the big ones, Roche and Eli Lilly, Novo Nordisk, they started decades ago. They started 8 years ago, 100 years ago, some of them even more than that. So very kind of like it's not the same speed as tech. And another depressing, maybe it's not a bit depressing if you want to, like as an entrepreneur in biotech. If you look at the market, the market capitalization that was created by Novo Nordisk and Eli Lilly in the past 3 years by a simple drug, GLP-1 based drugs, all this like the Ozempic of the world. It's more than the market cap that was created by all startups in biotech since 2000. And so it's very different.
It's incredible, and I mean, it speaks to actually a lot of things. You're right about there's regulatory capture and the fact that it takes so long. I think a lot of that is related to the balance sheet component. Like, it costs so much money to bring a GLP-1 to market. First, you have to run your phase 3, hundreds of millions of dollars, but then ultimately you have to commercialize it as well, which is billions and billions of dollars. And so what ends up happening is that the small biotechs do the innovation, and the large pharmas are basically corp dev and commercialization machines with very, very large balance sheets. And so it's a longer conversation about the sort of structure of biotech, but it does mean it's an interesting thing for someone like yourself, who's an entrepreneur in the biotech space. And as we described earlier, like, there is this challenge of to the winner go the spoils, and the winners are the people who actually commercialize the drugs. And yet they're not necessarily ones who are doing the innovating. Companies like yours are often innovating. They get a molecule, they sell it, it gets rolled up for a tiny fraction of what it's ultimately worth to the large pharmas. And I don't think you or I are going to solve that on our podcast today, but I think it is. It's certainly an interesting problem.
But you work in the field when you kind of like, you go home and you work for something that is for the really societal benefit, right? There is something that you don't work in gambling or gaming or porn or whatever. There are many you can do amazing companies in tech, but what's really the societal value of the company? So
I couldn't agree more. It's actually why it was about 10, 12 years ago that I made a career shift where a lot of what I'd been working on moved over into biotech and tech bio. And a lot of it was because you get to, if it works, you help cancer patients, or you help people with autoimmune disorders or lysosomal storage disorders, or whatever is the area that you're working on, or just to understand how it is that we work. So I'm going to pull on this sort of precision medicine piece, because obviously, part of what you're talking about is building a platform for people to build precision therapeutics in an mRNA space. And I think a lot of what you were doing earlier in your career with My Genome was helping people to, I'm going to call it's precision genomics of a different sort, maybe to understand diagnostically and, what are the causes of certain disorders. You'd mentioned earlier that, we work at Jimini Health in mental health, that there are, potentially genetic correlates and heritability of psychiatric and mood disorders. And I'm curious if that's an area that you spent much time on or have any thoughts about.
I didn't spend much time, but I was fascinated. There is an observation in human genetics that nearly all psychiatric traits are highly heritable. More than 50% of the variance of the risk between individuals is explained by genes. That's one observation. And you can parse it by twin studies. You don't need to do anything. Right. Just looking at twins. Another fascinating observation, which is, since I learned psychology as an undergraduate, when you look at twins, you can ascribe the variance to different components. So genetic is one component, shared environment is another component, it's like the household that you live in. And then the third component is typically we call it randomness. We don't know what to assign it. It's some unique experience in your life. Not genes, something random. So more than 50% goes to genes. Okay. Common environment explains typically 0% of the risk. Your household, when we look at these twin studies, variance between and inside twins, we don't get strong results from the household itself. No, I learned psychology. I learned dynamic theories of Freud, of looking at the parents and the role of the mother and the father and this. And when you go to regular psychological treatment, typically you speak about your parents, try to figure out things to help in your. But in the genetics, when you do these very quantitative studies that try to parse out, we don't see that effect. Still there is, so I don't know how to reconcile these two observations, but definitely these two communities don't discuss with each other enough, right?
I think there's a lot of effort now to help reconcile that. I will say, somebody who's lucky enough to attend, say, venture events where people who are working on cutting edge mental and behavioral health therapies, for example, there's a lot of talk about, say, what's happening inside the brain and what the downstream impacts are vis-a-vis your mind, and even things that sound a little bit hocus pocus or sci-fi-ish, like what's happening in your gut, which part of me, as somebody who lives in California, I'm like, come on, really? The microbiome? Yeah. But in fact, actually, the data are fairly robust, and most of your serotonin is produced in your gut. And so what you eat affects your mood is not, in fact, as sort of hocus pocus maybe as it sounds. And then you think about one of our advisors at Jimini is Dr. Seth Feuerstein, who actually I'll be interviewing here in the next few weeks, and he's a world expert on suicidality. And one of the interesting things that he talks a lot about is sort of the brain patterns of somebody who is suicidal. It's a little bit like somebody who's about to have a heart attack. It's not. It isn't necessarily sort of intrinsic to their long-term personality. It's sort of a state that the brain is in, in the same way that a dysfunctional heart can get into a sort of an incorrect state. And anyways, it changes pretty radically the way you think about curing it, whether that is with curing it with words, with therapy, or curing it with meds, or curing it with brain stim or whatever else. Yeah. And it's an exciting time, because I think a lot of the secrets that we just haven't had the right tools to unpack. We're starting to have the ability to do. It's still early though. I mean, it's very early, I think, for a lot of these problems.
And I think it's also exciting time because of this seems like as a pandemic of mental kind of like people struggling with mental issues right now, right? And who's that? Louis C.K. that said, right? Like everyone is amazing, but no one is happy, right? It's like that's kind of like we have technology, we can do like so many things, you know.
And he's absolutely right. I'm constantly struck. I recently reread Steven Pinker's book about enlightenment. I can't remember what it's called, but it's essentially about how like the world is astonishingly, incredibly, so much better than it was 300-400 years ago. The percentage of human beings, the length of we live, how much less pain we're in, how many most of us are free, live in democracies or something approaching a democracy, not in slavery, like educated, women's rights, health, like all these things so much better than they were 400 years ago. And we have an absolute epidemic of anxiety, depression, loneliness, suicide, et cetera. And the reconciliation of those can seem hard. I mean, I think part of it is human beings adapt very, very quickly, unfortunately.
Very quickly,
we adapt almost instantaneously to the fact that things are better. And then so there's our expectation level now. And so it's still easy to fall into anxiety or depression. So I'll give you another Louis C.K. joke about that, which was, he tells an amazing story. This is maybe 20 years ago about a guy who didn't know that Wi-Fi in airplanes was a thing. He walks up, he discovers that there's going to be Wi-Fi on the flight. And it's like, amazing. I didn't know this was possible. And then he gets on the plane and the Wi-Fi is not working and he's really ticked off. Which I think is a great encapsulation of like, human life can get better, but your individual mood, and this is true for me as it is for anybody else, may be sort of unaffected really by the macro stuff and is much more sort of malleable than you'd think. So fascinating time. You mentioned that the heritability of these diseases was over 50. How is that measured? You said twin studies. What are the ways that folks measure this?
It's very, very simple. You take monozygotic twins and they share 100% of their genome. And you look at the concordance of the trait between them. It can be height, it can be some sort of like a mental condition, it can be some breast cancer risk. Then you take dizygotic twins. Now, they have the same kind of like exposed to a similar setting of shared environment, right? They raised the same year, same kind of like same parents,
same parents, same
parents, but they share only 50% of their genome.
Right.
And you look at the concordance there of the trait. And you subtract the two concordances from each other, the monozygotic from the dizygotic. You multiply by 2 because you share half of the genome, so you need to account for half of it. And that's give you the amount of heritability that is ascribed to basically genetics. There are some nuances there, but that's the basic model.
Fantastic. This is so interesting. Well, I know we're going to come up on the hour here soon. So before we run out of time, I wanted to ask you a few questions. The first is, do you have, I love the breadth of your work, and I'm a dedicated generalist myself, and I'm just sort of insatiably curious, and I love to meet other people who are. I'm curious if there is a book or blog or podcast that you would recommend to people who are interested in AI and what's happening in omics and sort of the intersection thereof that you think would be really interesting for our listeners.
So I think it's not about AI, but I will recommend 2 books. One is it's called For Blood and Money. And this is how a specific class of drugs for blood cancer were developed. And the amount of serendipity and the inexperience of the guy that actually the CEO, his claim of fame before that, he sold the cookie recipe for McDonald's. This is how he made his money. And he was able to develop a new class of drugs. So it's an amazing story. And also explain how much money is important, but to raise money.
This is somebody who ended up with some cures for hematological cancers.
He actually, because his son died of brain cancer, and he thought that the drug that this company developed could help brain cancer, but then they realized they could actually pivot to a certain type of blood cancer. An amazing book, a page turn. That's the first one I would recommend. A second one, not related to AI, but I think actually it's pretty good just in general as a good book. It's the biography of Churchill. Again, not related, but it was a piece I lived in the UK for 2 years.
Yeah.
And it starts with basically with the Queen Victoria days, and it dies where Beatles had their first album, right? So it's a piece of history. I had to, in every page, I'm not a native speaker, but I had to look at a dictionary for at least once every page because the guy, he won the Nobel Prize for Literature, Winston Churchill? There is a reason. I learned so much just by reading this book and understanding the historical, and history explains how we ended up here today. So be able to actually dive and go with this book, which is fascinating and meaningful, explains quite a lot what happened in the Western world.
I love that. I feel like his life, if you want to look at one life during that roughly 80-year period where he lived, that would, for all the good and bad, and the glory of defeating the Nazis, maybe the horrors of having been something of an imperialist, and all of it encapsulated in a single life. I think that'd be
exactly. Gives you and it gives you context to today when you look at the world is kind of changing. I think it contextualizes many things. So I really, so it's not about AI. So if I disappointed,
but no, not at all. That's fantastic. And in keeping with somebody whose interests are very varied.